Um, Evolution is kind of I think is an interesting one. I like talking about it. We tend to really think about evolution from a human perspective. And it's one of the things that kind of grabs the attention a lot, human evolution. And it also tends to draw a little bit of some more passionate responses with human evolution. Human evolution, remember, is just one part. It's one example of evolution. It's not what evolution is all about. It's just one aspect of that. And so we're going to be looking at then how, we're going to be looking at what are the mechanisms that are driving this process of evolution, okay? So evolution is really, it's the change in characteristics of species over several generations. And evolution itself is really very factual. There are many examples that we can see how species have changed over time. And that's really all evolution is. So I'm kind of giving you this overview of what we're going to be looking at. Here's just one example of how the horse from 50 million years ago was a rather small organism, and to where we are even today, bigger than 3 million years ago, how the horse has grown in size, it's changed, its bone structure, while has remained fairly consistent, has just grown larger to meet the growing demands of the growing horse, to fit the structure for that. And so this is just one example of how there's been change in characteristics of a species over time. And I also love kind of talking about the giraffe. I think the giraffe is always kind of the interesting one, is that, remember, the giraffe's neck has not always been long. That the giraffe actually had a short neck and when it was first seen, and the giraffe's neck has gotten longer over time. We can look at almost any organism and see how they have changed over time then. And that's this idea of evolution. Now, how do we get to evolution? And how does then this idea of change over time work? Well, it comes about this idea of natural selection, which we're going to be talking about tomorrow, and this idea of descent with modifications. So really what we're looking at now in this idea of descent with modifications is we're looking at the change in the gene frequency within a population over time. Now again, gene frequency gets back to this idea then of, there we go, I'm gonna go there and go here. Remember, this gets into this idea of gene frequency of genotype. So we're directly going back to the last unit we just got done learning about with Ms. Klepper with genetics. So we're talking about now how alleles, right, are getting passed down from generation to generation, how genotypes then affect phenotypes and the expression of those traits. And so this idea of descent, right, that we are inheriting traits, and those traits then are being modified. And that modification is what we're going to be looking at. How then are traits being modified? And that's what this whole idea of evolution is going to be looking at. We're going to be starting that today by looking at this idea of how do we modify the genotypes that are being inherited from generation to generation. Our first topic for today is going to be looking at mutations. Then we're going to be getting into natural selection and sexual selection. We're going to be getting into speciation. We're going to be also talking about mimicry and camouflage. We're going to be talking about how those genes are actually then, how they can actually change through the population over time, microevolution and coevolution. So these are our topics that we're going to be covering in our next 10 days in our unit. This example here, this picture here, this is actually a fairly complicated one. And really what it's showing is that as we start early, so this is going back in history and working our way forward, we can then see, and we can actually really see, but these are changes to the genes. These are genetic changes. And then at each level, so from A to B, from B through the process of change, we created the cow. At C here, we get the hippopotamus. So we get a land mammal. We get an aquatic mammal as well, or terrestrial and aquatic mammal. But then through the process of change, we can see that we get then again terrestrial organisms. And through the process of genetic changes, we get to today's whale. And this is one thing that we actually do know, is that our aquatic mammals, so things like whales and dolphins, actually evolved from terrestrial organisms. We know that life then started in the water and it came out onto the land. And from once it got on the land, it then radiated out to all the different things that we start to see. But then the aquatic mammals went from the land back to the water. We know that for a couple of different reasons. We can see it from a genetic perspective. We actually can look at the way that whales swim is in an up and down motion, which is the same way that a wolf's spine compresses and a dog's spine compresses when they run. So we're seeing very similar structures, anatomical structures, and movement structures in terrestrial mammals to our aquatic mammals. So these are all pieces of evidence that we use to see how then this idea of change over time takes place. So any questions about the overview before we get started with things today? My goal every day is to give you guys a half hour of work time. So that means my lecture will be done no later than 11 o'clock this morning, okay? After that, I want to give you a half hour of time for you to be working on every day's homework assignment so that you're getting plenty of time to work in class, ask questions, and hopefully even get the assignment done in class, okay? Because these assignments do get a little bit longer, and they're going to take a little bit more time, especially kind of our upfront ones. Today's, tomorrow's assignments in particular take a little bit longer to do, okay? So here we go. Let's take a closer look now at our first mechanism. We're going to be talking about mutations today, and how then mutations play a role in this idea of change over time. All right, so here we go. Our essential question is, what are the mechanisms which drive evolution? Our vocabulary words, we've talked about evolution, change over time. We're going to be talking about a mutation and what is a mutation, and then the different types of mutations. But before we get into mutations, we actually have to go back in time a little bit. We have to go back to first semester. The last time I taught you all this, I was teaching you about DNA replication, and then making proteins. And now all of that, what I taught first semester, actually relates to the last topic that we learned about genetics. So remember our central dogma that we were learning about first semester, that the fact that DNA makes RNA, which then makes amino acids. And without the idea of DNA to RNA to making the amino acids, which are basically proteins. Now remember, proteins then are the expression of a phenotype. So remember, a protein is things like hair color and eye color, skin color, your ability to roll your tongue, your ability to break down a sugar in milk, are all because of the expression of a protein. And you get that genotype from your parents. Remember, you are inheriting one allele from mom and one allele from dad. That goes back to Ms. Klepper's genetic unit. And so in this process, remember, we've got our DNA model, which is double helix in shape and structure. We've got our base pairs, cytosine and guanine, adenine and thymine, our 4 nitrogenous bases. And we get C and G pairing together, and we get A and T pairing together. Those make up then the backbone, or our steps, of the DNA. And then we've got sugar and phosphates, which are connected to make our backbone. Remember, through DNA replication, we actually then unzip the DNA, make more DNA by adding new DNA nucleotides to the unzipped portion. And we're going to talk about then what's taking place during that time, because it does relate to today. And then we make more DNA. Then through the process of making the RNA, through the process of transcription, remember, when we transcribe something, we're taking the message of the DNA and we're going to transcribe or write it down into the mRNA. That's the messenger RNA. Remember, that's taking place inside the nucleus. So where we're going to read then the DNA line, and we're going to make that into the mRNA line. And that mRNA line, remember, is going to be read in codons. So we would have then our 3-letter sequence of our codons. And we can see that again down here below when we then go through the process of translation, that once the mRNA codon leaves the nucleus and goes to the ribosome, through the process of translating it, we're going to translate the mRNA codons into then the polypeptide, and really what we're doing is translating them into amino acids. Now, it's this amino acid sequence, glycine, remember our amino acids that we're reading our codons with, and our codons, remember, are being read by our amino acid wheel, where we, again, use the mRNA codons to read the amino acid wheel. Remember how we read this is our first letter is on the inside, second letter is on the second ring, and the third letter is on the third ring, which then leads to your amino acids, which are on the outside ring. So just as a practice, remember, if you get GCU as your mRNA codon, that we would go then G is our first letter, C is our second letter, U as our third letter, and that GCU codon codes for the amino acid called alanine. And so we would write down alanine as our amino acid. Remember, when we link the amino acids together, here in our picture, that when we link these amino acids together, this then becomes our protein, which then becomes the expression of the genotype. So again, hair color, eye color, ability to roll your tongue is all because of your amino acids or your protein sequence. So today we're going to be talking about then is with this idea of mutations, right, is if we change some part of the code. And one of the places that we see a lot of those changes taking place is actually up here. If we change this adenine to, let's say, a guanine, a G. So the process of DNA replication, one letter or multiple letters change, right? We call this part then the mutation. And then subsequently, what does the mutation do? What's the effect that it has upon then everything else past that point? So if we get the mutation at the DNA level when we're doing the DNA replication, remember that change here to guanine would be then changing this to cytosine. Let me use a different color because I realize that just doesn't quite work. That would change that to cytosine, which would change this back to guanine, which would then change this, and I'll just change this to lysine. And so now we've changed the amino acid sequence. When we change the amino acid sequence, we change the protein. If we change the protein or the amino acid sequence, we change the function of the protein. If we change that function of the protein, we change the expression of the protein. So we change the hair color or the eye color or the skin color or the ability to roll your tongue or that ability to break down sugar and milk. All of those different examples are because we change the expression of the protein. So today we're going to be looking at then how do these changes come about? Okay, we call those changes then mutations. There we go. You can all see that now. A mutation is a change in the DNA of an organism. The mutation changes the DNA, which changes that mRNA codon, which then changes the amino acid sequence, which then changes the phenotypic expression of the genotype or the phenotypic expression of the protein. So again, we change the structure, we change its function. We change the function, we change the way that it's being expressed or used. So again, a lot of times these mutations are taking place in DNA replication. Well, we tend to actually, these DNA things will copy incorrectly at times naturally. We're going to talk about it in just a second there. But this idea that mutations can be beneficial, they can actually help. They can create an advantage for the organism, what we call their increase the fitness. And fitness is really just talking about and organisms ability to reproduce. So as an organism, if it increases an organism's fitness, it means they're able to live longer, to reproduce more. So a mutation can be beneficial that then allows that organism to live longer, to reproduce more, which then to pass along its traits more frequently. We can also then get a beneficial mutation result because it increases variation. Now remember, variation is the way that we look differently from other people, from other organisms of the same species. We all have different hair color and eye color and skin color. We're all different heights of tall and short. When we think about giraffes as an example, that variation of neck lengths, right? That we've got some necks that are longer, some necks that are shorter than others. Legs that could be longer or legs that could be shorter. So we can see variation built into any species. So this idea of a mutation can actually change and increase that variation, which is a benefit to not only the organism, but also to those species. You can get a neutral change to a mutation, the fact that it does not increase an organism's fitness, right? It doesn't help them to live longer and reproduce more, but it also doesn't hurt them to live longer, reproduce more, right? It has a neutral effect upon it. And we'd also say that mutations could be more harmful. This is the one that we tend to think about when we hear the word mutation, right? Is we tend to think that mutations are bad, that it creates a disadvantage for the organism. It decreases their fitness. And when we get then mutations that are harmful, we tend to see then that these organisms then die off. And it's these organisms then that become extinct. Extinct, T-I-N-C-T. There we go, okay? And when we get those harmful mutations that don't give an advantage, then the disadvantage, those ones are being outcompeted. They have a decreased fitness. It might make them not as camouflaged. It might make them slower. It could make them not as fast, maybe not as fly as far, whatever, which then would harm them and they would die off and become extinct. So we can see many examples in our fossil record where we're finding then changes that were not beneficial to organisms. So what's causing then a mutation? Well, DNA fails to copy accurately, and this occurs naturally. Again, this is occurring, right, in DNA replication. DNA replication actually has errors in it, and we correct those errors about 99% of the time, get corrected. There are instances where there are natural changes to the DNA sequence, but those get corrected through the process of, actually it's DNA polymerase number 3, which goes back through and it proofreads it every time the DNA gets copied. So we can see many examples of that, right? You and I, everybody in this class, right? DNA has been copied correctly many times over, and those mistakes then have been caught, and we catch those many times. But there's also then other external influences that can create mutations. Things that are caused, mutations are caused by exposure to specific chemicals or radiation. Those things that could be sunlight actually causes then mutations to occur. Those mutations then come about through the process we call cancer. Cancer is a change in the DNA sequence. It changes the information, and it causes then the DNA to continue to keep replicating. It doesn't stop, which is a change in the genetic information, which then causes tumors and things like that. We also see that X-rays, radiation, is probably one of the more common and more frequent ones that we see when you go to the doctor, when you go to the dentist, and they take an X-ray. The X-ray tech is the one who's leaving the room to go stand behind a lead wall because they don't want to be exposed to those X-ray radiations, When you go to the doctor or the dentist and you're getting an x-ray, they generally would put a lead apron on you. And if they don't, you should be asking for one. Because exposure to x-rays is one that can very readily break down. And actually what it does is it breaks down the bonds, the nitrogen bonds in your DNA, and allows then for the exchange of adenine and thymine, cytosine and guanine to take place and not correctly bond then. So there's a lot of things then that can create this idea of mutations, which is why especially this one of skin cancer is an important one, right? You wearing sunscreen going out, because that prolonged exposure, years and years and years of being out in the sun can create skin cancer, which is just a type of mutation. So when we start talking about mutations, then where are they? And mutations can occur in two different places. One, we call those the somatic cells. This goes back to our genetics unit with Ms. Klepper. Remember, soma means body. So we've got our somatic cells, which are our body cells. So we're talking about things like skin, hair, eye, lung, heart, muscle tissue, our somatic cells. When we get a mutation that takes place in a somatic cell, we don't pass along that mutation to next generations. It's only affecting that one individual, right? It's not changing it at a level where you're going to be passing along that trait to the next generation. When we get mutations that take place in the germ cells, remember the germ cells are either the sperm or the egg. When you get a mutation that takes place in the sperm or the egg cell, this one does get passed along to new generations. Because remember, it's the sperm and the egg that's creating your offspring. So if we change a mutation in the sperm or the egg, that then will then get passed on to new generations. How do mutations happen, Quentin? Well, mutations again can be random, right? Mutations again could be through exposure to sunlight, x-rays. X-rays is kind of the big one when we start talking about germ cells, right? But a lot of times these things, especially when we're talking about germ cell mutations, actually is just more of a natural process than anything else. Remember, there's a lot of things that go right in this process, but there's also always going to be exceptions to those rules that go right. And so when things do go wrong, and again, especially when they go wrong at a germ cell level, we tend to see a little bit more disastrous. There tend to be the effects tend to be a little bit more severe. And again, they get passed on to that next generation because they're found in the germ cell line, not the somatic cell line. All right, so that sets the stage for us for mutations, right? Which is really now we've talked about, right, where they can be found, how are they being done through DNA replication, right? It's going to get just a change in the DNA sequence. So, right, this is kind of a great example of that. Here's our original strand of DNA, and then there's our mutant copy where we change the purple one to a yellow one. And again, this is just an example of taking a T and let's say changing it to a C. Okay? So it's not pairing it up correctly. So now we're going to be looking at then what are the different types of mutations. And this kind of gets into now the heart of our lecture for today, is what are the different types of mutations that are taking place. So there's two main types of mutations. The first ones that we're going to be looking at is what we call a point mutation. And in a point mutation, we are changing one base in the DNA. It's one point that's being changed. So we tend to think of these types of point mutations as what we call a substitution mutation, because we're exchanging one base for another chemical letter, such as switching adenine to guanine. So in this type of point mutation, instead of adenine pairing with thymine and cytosine pairing with guanine, and I'll even actually put the bonds there so we can see those correctly, what would happen in that type of mutation then is you might get adenine pairing with cytosine or thymine pairing with guanine. And so we tend to see then those are the types of mutations. Again, this is a point mutation where we're changing one letter in that sequence of the DNA. Now, what are the effects of changing one letter in the DNA sequence? Well, there's 3 different effects to that. The first one that we're going to look at is what we call a silent mutation. And in a silent mutation, there is a change in the DNA sequence, but there is no change to the amino acid. Okay? So no change to the amino acid. So as the example down below here, here's our original strand of DNA, that 3-letter sequence of our DNA called a codon, or TTC. That mRNA codon then is AAG. The transcribed DNA makes AAG. And then when we translate the mRNA codon of AAG into an amino acid, we get lysine. Now, in the silent mutation, instead of having TTC as our original strand, we get TTT. That changes the codon to AAA, which changes, and actually it doesn't change, that's where this silent part comes in, is it changes AAA, the codon still codes for the same amino acid of lysine. So there's no change in the expression, but there is a change in the letter. So there actually still is a mutation, but in this mutation there's no change in amino acid sequence, right? So there's no change in the expression. The second type of point mutation that we can find is a nonsense mutation. And in a nonsense mutation, again, we're going to get a substitution, a 1-letter substitution, where the change in the DNA sequence then makes a stop codon. And that stop codon then changes everything behind it. That once you get the stop, remember, if we go back to our ideas of introns and exons, this goes back to the first semester again, that the exon will always be between the start and the stop. So if we now get a nonsense mutation where we're creating a new stop codon, that means we're losing all of the information behind the stop. So depending upon where this nonsense point mutation is, you could lose a lot of information or a little information in this process. So as again we see down below here in the nonsense mutation, we have our original strand of DNA, TTC. Our nonsense mutation, now we change the first T to an A, ATC. That changes the mRNA codon to UAG. And UAG then on the amino acid wheel codes for stop. So again, everything behind it would then change. And so the nonsense mutation is actually one that can have a fairly major impact because it's changing the entire thing behind it. A silent mutation doesn't really change anything. Nonsense mutation changes a lot because it changes to a stop codon. Our last example then is a missense mutation. And in a missense mutation, there is a change in the DNA sequence. And what it does is it changes the amino acid. Let me go back and just highlight that part. Changes the amino acid. Well, if it changes the amino acid, then it changes the function of the entire protein then. Because we've changed the sequence, we change the function. If we change the function, we change the expression. So with our missense, again, our original strand of DNA of TTC, a missense, we get TCC, as that then creates a new codon of AGG on the mRNA, and then mRNA codon then codes for a new amino acid of arginine. We could also see again another one of TGG or TGC, could make theranosine. So instead of it making lysine, it makes now a different amino acid, which again changes the function and it also then changes the expression of the protein. So remember, if we change the expression, that's like changing the hair color, the eye color, the skin color, your ability to roll your tongue, your ability to break down sugar in the milk. Okay? So point mutations, a one-letter change, generally found in the DNA, which then changes the mRNA, which then changes the amino acid, or possibly changes it, except for the silent mutation where it doesn't. All right? So that's the point mutation. Now, the other type of mutation that we find then is what we call a frame shift mutation. And in a frame shift mutation, we're changing the reading of the codon. It's actually changing how the three-letter sequence is getting read. So it's when the gene or codons change, and this changes the amino acid sequence, and it changes the function of the protein, and it again also changes the expression. So again, when we change the expression, again, this can lead to greater variation, right? This could lead to a higher fitness. So again, these mutations aren't necessarily bad things. So don't forget that part. When we talk about a frame shift mutation, if we're going to be changing the way that the codon is read, we can do that through two different ways. We can either do that by an insertion, where we are adding letters or codons into the sequence, or we can do it through deletion, where we're losing letters or codons from the sequence. So as you can kind of see as a quick example here with the insertion, if this is our original strain of DNA, CTG-GAG, and we insert a whole new codon of TGG, when we read now the new sequence to make new codons, we get the first codon of CTG, which is the first one. We get the second one now of GTG, which changed. And we get the third one now of GAG. So again, we've changed and added a whole new codon, but you also could just add a letter to it as well, which would then change the way the codons get it grouped after the insertion. Likewise, too, with deletion, that if you look at the way that these codons then get read or grouped together, that would be our first one of then G or CTG, and the second codon of GAG. But if you take away, delete the two G's, you now get a codon of a codon of, we'll change the color then, of CTA, which is different from the first two. And we'd have to see then what is actually going to be past the G there, because we don't know what it is. So when we insert letters or codons or delete letters or codons, it changes the whole codon sequence then. Okay? I like the examples here that I've got at the back part here when we start looking at how we read sentences. And as we read then these sentences, the fat cat ate the rat. Remember, these are all letters or words in groups of 3 letters, which represent the codon, right? So this here, and actually let me use the highlighter because these are a little bit better then, right? Is that the and then fat and cat and 8, right, are all codons, 3-letter sequences. And when we insert one then, the fat, and we change then that C to an H, actually we don't change it, we insert an H in, right? And then we still keep groups of 3. We now get HCA, TAT, ETH, ERA, and T. So we've changed everything behind it. So all of this then gets changed because we've inserted that new letter into it. Likewise, too, with a deletion then, when we delete the H out of it, and then we combine the other letters remaining, it's still in groups of 3, it changes the entire meaning of that sequence and subsequently the expression behind that deletion. So these idea of gene mutations tend to have bigger effects because you're changing, right? There are more amino acids being changed. When we talk about these idea of frame shift mutations, this idea of insertion and deletion, you still can get a fair significant change, right, when you look at this idea of the missense or the nonsense when we get to the stop, right? So this one still has could still have a fairly big impact depending upon where that 1 point mutation is. But silent and missense tend not to have a big impact or change upon it because you're only changing one amino acid, but it only takes one to change the whole process. So, but the frame shift ones tend to be bigger in their effects. So, that's kind of my lecture for today on mutations. So we've seen now where they're found.